Angiogenesis, or the process of blood vessel formation, is involved in both pathophysiology and physiology. In physiological conditions, we see more of blood vessels coming up during wound healing, and in pathophysiology, during tumors, during cancers, we have blood vessel formation. And from last 6 years, we have been looking how this process of angiogenesis is related to liver diseases. And in the work which we have presented this year in ASLD, we have seen angiogenic mechanisms, angiogenic genes in the pathophysiology of non-alcoholic fatty liver diseases. So we have used 3 different approaches. We started with the differential gene expression studies in the patients, steatosis, NASH, and the controls. These were the 3 study groups. And we did a microarray analysis, high throughput gene expression study, and then we selected around 25 to 26 genes which were associated with oxidative stress and angiogenesis. And then for validation of the genes, we selected a higher subject group of around 150 subjects, where around 40-50 patients were of non-alcoholic fatty liver diseases, which included mild steatosis, and around 40 to 50 patients were of severe steatosis, which is NASH, and 40 controls which were healthy. And we validated the angiogenic genes in the subject group and found that around 13 genes are differentially expressed between various subjects. And we did a bioinformatics analysis of these genes and studied what kind of transcription factors are controlling these genes. Like we wanted to study the pathway, the mechanisms which lead to the upregulation or downregulation of these genes. And the next approach was to take up some in vitro studies, where we have co-cultured steatotic hepatocytes with endothelial cells, because angiogenesis is a property of endothelial cells. So we made an in vitro model where we incubated the hepatocytes with the endothelial cells and then studied the angiogenic functions of these endothelial cells, and also studied the gene expression and just correlated whether some of these genes are correlating with our gene set, what we found in the patients. And the results say, as far till date, that definitely there's an increase in angiogenesis, there's an increase in the endothelial cell proliferation with more fat, with more steatosis in the patients. And we have landed up onto some genes which are really important, and now we are looking at how these genes are regulated and how these genes they actually lead to steatohepatitis.